My name is Ram Dixit. I'm Associate Director of Education in the CEMB, and my pleasure to welcome all of you to this event. So I appreciate you taking time here in person, and for those of you who are on Zoom to join us to hear about and celebrate all of the achievements and wonderful research that our summer undergraduate researchers in the CEMB have spent time doing the last 10 weeks or so. So students, I hope you enjoyed the program. I hope you enjoyed your individual research projects, even if I've no doubt when it pushed you outside of your comfort zone, and that's partly intentional, if not completely intentional. Besides diving into research, I hope you took time to make new friends in your lab, outside of your lab, and take some time to have fun and explore WashU and St. Louis. So hope you had a good summer. And remember that we are now forever part of your scientific network, all right? And so I encourage you to stay in touch with your lab, with your bench mentor, with your PI, with your Bayer mentor. We are here to support you. We love, all of us, love to hear about what you're up to as you go forward and your achievements and, as you progress in your career. So stay in touch, okay? Yes, a couple of things I'll say. One is we also wish you all the best with your applications to the BMES conference. And as you know, if you're selected, CEMB is very proud to support your participation at this conference. So all the best. I guess you guys will probably hear about them pretty soon. Okay, next week. Okay. And then to wrap up here, I want to keep my remarks short. Really want to thank both Patricia and Gwen. They're the ones who do a lot of work, before this program starts, all through the program, with logistics, planning, executing the program, everything that really goes into it. So this would not be possible without them. So please, a round of applause for both of them. Yeah. Let's also thank your bench mentors and your bio mentors who took time and to train you and mentor you, and mentors know that your trainees appreciate all the time and effort you put into this. So again, thank you to the mentors. All right, so then without any further ado, I welcome Patricia Widder to the podium here. She is the CEMB Education Program Coordinator, and she'll get things rolling in a minute. Patricia.
Welcome, everyone. It is my pleasure to open the presentations of our student work. Throughout the summer, the students have been doing not only research work, but also external educational programming that we've worked on together. We've talked about graduate school. We've talked about scientific presentations. We've talked about professional development, how to make a good poster and how to make a good slide, how to write an abstract, and how to submit your work to conferences and scientific publications. And the students have done so well. I'm very, very proud of you. And it has been my pleasure to work with each of you this summer. So let's go ahead and begin. Our first presenter today is Abria Granger from the University of Texas, San Antonio. She's been in the Fuzhong lab, and her presentation is called Development of Strategies for Targeted Delivery and Retention Enhancement of Mitsugumin 53 for Tissue Regeneration. Let's welcome Abria.
Got it. Hi, everyone. My name is Abria Granger, and as Patricia mentioned, I was in Dr. Fuzhong Zhang's lab this summer. And I worked also with my lab mentor, Zhang, and my project is on the targeted delivery and retention enhancement of Mitsugumin 53 for tissue regeneration. And here is an overview of my presentation and poster. So to start off, Mitsugumin 53 or MG53 is expressed in skeletal and cardiac muscles of human, rats, and mice. It plays an important role in muscle repair and regeneration after injury. It's an essential component of cell membrane repair machinery, and unfortunately, the protein doesn't last very long in the bloodstream after injection. So it has a short half-life, and it doesn't have enough time to reach the injury site to start regenerating the tissue or cells. So to enhance the retention time of MG53 at specific injury sites, our strategy is to genetically fuse MG53 with specific cell targeting peptides so we can enhance the retention time of MG53 at specific injury sites. And the recombination of the MG53 and cell targeting peptide could be used for therapies and drug delivery applications that aid in tissue repair and regeneration. Here are the cell targeting peptides. IMTP, or ischemic myocardia targeting peptide, CTP, cardiac targeting peptide, 5X ELP SpyTag, elastin-like peptide, and SpyCatcher IMTP. So SpyTag and SpyCatcher are two peptides that can attach together, and they form a covalent bond that can fuse different proteins together. And specifically, it provides stability and ability for different proteins. And it also helps with like the versatility when you combine and assemble different proteins together. ELP is added to MG53 5X ELP SpyTag because SpyTag can't combine with MG53 by itself. So here's the protein and gene configuration of each of the recombinant proteins. So there's the MG53, IMTP, MG53 CTP, and you can see the MG53 5X ELP SpyTag is first, MG53 is connected to ELP and then SpyTag, and then SpyCatcher IMTP can bind to the SpyTag and form a larger recombinant protein. The first step of doing protein production is E. coli cell culture. So I take 1 liter, this whole process is done in 5-liter batches because of low protein yield. So I take 1 liter of LB media and I add a seed culture, which is just LB media with the E. coli cells and the protein added into it. And I add 1 milliliter of antibiotic. And then it is shaken and incubated at 37 degrees for about 4 hours. Then after that process, I check the optical density to make sure that it's around 0.8. And then I can induce the cells with IPTG, which basically just starts protein expression. And then after that, I can put the flask back to incubate at 16 degrees for 4 hours. And then after that, to harvest the cells, I centrifuge the media and I resuspend the pellet in PBS and freeze it for protein purification. And protein purification. So the cell lysis that I previously did in the E. coli cell culture, I thawed out and I ultrasonicated to release the protein from the cells. And then it's centrifuged again so that I can take the supernatant and load it into a nickel column for IMAC. So IMAC, or immobilized metal affinity chromatography, is done on the FPLC, which is the fast liquid protein chromatography. So that process just makes it easier to monitor the elution of the protein, and it makes the process quicker because all of the solvents are already attached to the machine. So during this process, the protein is put into the column and it binds with the nickel inside the column, and the rest of the supernatant flows through. And then during the elution process, the bond between the nickel and the protein is disrupted and the protein is released through the elutions. And then with the elutions, we make an SDS page gel to check for protein expression. And based on the results that we get from the SDS page gel, we can collect certain elutions that has a higher concentration of the protein. Then it's put through a dialysis process to get rid of any impurities like the imidazole used in the IMAC. And after that, it's put in a centrifugal filter unit so that I can concentrate the protein to about 2 mg per mil. And I check the concentration of the protein with the nanodrop spectrophotometer throughout the process. And then after that, it's frozen with liquid nitrogen and dried out with the lyophilizer. And here are the results, the SDS page gels. So this is a batch of the MG53 IMTP. So here is the protein marker, so you can estimate the molecular weight of the protein. This is the band that we're looking at. So you can see right here that 55.91. I should mention that I think this shifted, so 50 should be here instead of up here. But 55.91, so you can see that this is around 55.91, so this is our intended band. And basically the intensity means that there's a high concentration of protein and protein expression here. So we collected elutions 24 through 43 for the protein. And here's another batch of MG53 IMTP. So we collected 25 through 50 for this one. And then here's Spycatcher IMTP. So this gel is on a 15% gel, which is why it looks so different. And Spycatcher IMTP has 10 kDa, so it's a lot lower molecular weight. So we ended up collecting 8 through 51. And here's a batch of MG53 CTP. The expected molecular weight is 56.39 kDa. And you can see here that we collected 101 to 180. And here is the results for my concentration and total yield of the proteins that I was able to get. So for MG53 IMTP, our targeted concentration overall was 2 mg per mil. So I got 2.074 for MG53 IMTP and for 2.052. And the total yield was 7.549 and 8.824. And here are my conclusions. We were successful in the production and purification of MG53, IMTP and CTP, and we were able to determine the protein expression and molecular weight by analyzing the SDS page gel images. We were also able to successfully obtain the targeted concentration and total yield of MG53 IMTP and MG53 CTP. Unfortunately, we're unable to finalize the purification process of MG53 5X ELP SpyTag and Spycatcher IMTP due to time constraints. And additional testing with these proteins for degradation and cell targeting properties could be done in rats and mice. And these results could provide advancements in using the proteins for regenerative therapies and drug delivery applications. Thank you. Are there any questions? It's degraded after, I guess, like in the blood circulation. So after you inject it, it doesn't have enough time to reach. Yeah. Yeah.
Exploring if
Yes, exactly. My project in particular doesn't deal with, I guess, the second phase. Our lab does have a collaborator, but I think it could be applied to a hydrogel because if it's an injectable or anything, so you could try to use a hydrogel to encapsulate it and release it into like a rat or a mice to see if that works. Thank you.
Thank you so much, Abria. All right, our next presenter is Ross Aiello from Ohio State University. He's been in the Amit Pathak lab this summer, and the title is Examining the Role of Hypoxia in Extracellular Matrix Remodeling and Cell Motility in the Context of Breast Cancer Metastasis. Let's welcome Ross.
Use the laser. Hello, everyone. My name is Ross Aiello, and I am a rising senior from The Ohio State University. And today I will be presenting my project on examining the role of hypoxia in extracellular matrix remodeling and cell motility in the context of breast cancer metastasis. So first, a little bit about what is hypoxia and the importance of breast cancer. So breast cancer is incredibly prevalent in today's society. It is responsible for 670,000 deaths in 2022 alone in just the US. Additionally, hypoxia is a common hallmark of breast cancer and results because of the abnormal cell proliferation and abnormal angiogenesis and vascularization of the tumor, which results in certain parts of the tumor not receiving as much oxygen as it should. Additionally, hypoxia is a low oxygen concentration, which is typically around 1.3% for breast epithelial tissue. This is compared to 8.5% for normoxia, which is what it is normally at, and 20.9% for the atmospheric concentration. And hypoxia is important as it is shown to induce many different pathways that are shown to increase extracellular matrix modeling, which then enhance the cellular motility that allow the primary tumors to metastasize and decrease patient outcomes dramatically. So understanding hypoxia and its effects on the tumor microenvironment could possibly lead to novel methods for cancer diagnosis and treatments in which hypoxia can be used as a diagnostic tool or could somehow be targeted to stop the development of the tumor. However, there is a problem in which the interactions between hypoxia-promoted ECM remodeling and cellular motility in breast cancer are not entirely well characterized, even though it is a area of active study. So our experiment seeks to solve this problem by the following experimental setup. So first, we have collagen, type 1 collagen that was remodeled by both tumorigenic and non-tumorigenic epithelial cells, otherwise known as BT549 and MCF10A cells, under hypoxic, which is again 1% oxygen roughly, and normoxic, which is roughly around 8.5 to 21%. And single cells were seeded and tracked for 24 hours following the collagen remodeling, and then the cell morphology, motility, and resulting collagen remodeling was imaged and analyzed. And diving deeper into my methods, I first created a 24-well plate as shown here using PDMS, and then we treated the well plate with glutaraldehyde to increase the collagen adhesion to the plate. Following the treatment, we coated the wells with 2 milligrams per milliliter of collagen and allowed it to crosslink. Following the crosslink, we seeded a first round of cells, which included a sham, which is no cells, and BT549 cells, which are cancer cells, and MCF10A cells, which are healthy epithelial cells, and allowed the cells to remodel the collagen for 2 days under normoxia, which is like a standard Thermo Fisher incubator, and hypoxia, which was a special incubator, otherwise known as a 2-gas incubator. And following the 2-day collagen remodeling, our first round of cell seeding was removed via trypsin and gentle rinsing to make sure that the structure of the collagen was preserved. Following the removal of the cells, we seeded a second round of cells, which included single cell seeding of MCF10A healthy cells. And then we gave time for the cells to adhere and then transferred the well plate to a time-lapse imaging microscope where the cells were tracked or at various positions for 24 hours. Following the tracking, the well plate was removed and the cell positions were analyzed using a variety of MATLAB, ImageJ, and CellPost scripts, and the collagen was imaged using reflectance imaging. So this then leads to our results, and our first result is that hypoxia affects extracellular matrix remodeling behavior in 2D, but for different cell lines, but different cell lines have a different response to hypoxia. So we can see in figure 2A and D that these are the sham groups, which do not have any remodeling prior to single cell seeding and cell motility tracking. So the collagen is decently dense and has a good degree of cross-linking. However, the two cell lines respond differently to normoxia and hypoxia. For normoxia, the cancer cell line produced a significant amount of degradation as shown in B here, which leads to a lower overall signal and reflectance imaging and a higher alignment. However, in normoxia, the healthy cancer cell line, remodeling increase and collagen deposition also increased, leading to a denser image as shown in this figure here. However, in hypoxia, the results are switched, in which for the cancer cell line, the cells increase remodeling and collagen deposition, which leads to a denser collagen as shown in this figure here. And the healthy MCF10A cell line increased degradation, which leads to a less dense image as shown in figure 2F here. And however, both cell lines did show an increased alignment of the collagen, which then leads to our next result in motility. That hypoxia remodeled extracellular matrix increases migration distance and persistence. So we could see in figures 3A through D that the green plot right here is the hypoxia group. And we can see that the distance and the persistences are shown to all be higher for the last hour of cell tracking compared to the other two groups, including normoxia remodeling and the sham group. And this is largely because of hypoxia showed an increase in collagen alignment. So the cells can travel in a straighter line and much farther than others remodel collagen in 24 hours. Additionally, I should add that persistence is the linearity of travel. So a higher persistence would mean a straighter line of travel. It also ties into our next result that hypoxia remodeled extracellular matrix increases migration velocity only for the cancer cell line. So we can see in figure 4A here that the red group, which is normoxia here, has a much higher velocity compared to the other two groups, which are yielded statistically insignificant. However, for the cancer cell line, as shown in B here, the green group, which is hypoxia, has a much higher velocity compared to the other two groups. However, it is also worth noting that for the MCF10A healthy cell line, the upward trend of the hypoxia group is much greater than the normoxia group. And there is this lagging period right here for the starting time point. This could be due to a variety of reasons, including that hypoxia may have had some unintended effects with cell collagen adhesion, which leads to the cells needing longer to adhere to the collagen. Or it could also be explained to the fact that as time went on, the time to set up experiments decreased, which led to less time for the cells to adhere to the collagen before the experiment was started, as the cells had already begun to migrate in the normoxia and the sham groups before the hypoxia group could get started. However, as with everything, more experiments should be performed to verify these results. Moving on to the discussion and the conclusions, summarizing my results. Hypoxia appears to have produce increased collagen alignment, resulting in increased migration distance and persistence for both of the healthy and the cancer cell lines. And I'm wondering if it is due to hypoxia-inducible factors such as HIF-1 alpha. And a future experiment could be exposing tumorigenic and non-tumorigenic cell lines to hypoxia and measuring the difference in HIF-1 alpha expression and understanding how hypoxia affects these different pathways that produce these different collagen remodeling results. Additionally, our next result is that there is a significant difference in invasive cell collagen remodeling between normoxia that produce extensive degradation as shown in B here, and hypoxia, which produce more remodeling as shown in C here, which reveal that tumorigenic cells could possibly rely on hypoxia for metastasis enabling remodeling. However, this is a broad claim and more experiments should be performed to verify it. And I have a hypothesis in which, so we have a tumor and the cells on the border of the tumor, like with the interface with the tumor and the native tissue, have a higher oxygen concentration due to it receiving more vascularization from the native tissue, which leads to a remodeling behavior as shown in B, in which these cells degrade the matrix more. So this increased amount of matrix degradation leads to the creation of new pathways for the cancer cells to travel and eventually enables metastasis, such as the travel of the invasion of a blood vessel, which carries the cancer cells away and metastasizes. And additionally, the center of the tumor, which does not receive as much vascularization, has a hypoxic environment, which leads to the remodeling as seen in C, in which the matrix is much denser, which then enables the cells to move around more and eventually escape the center of the tumor and go into these newly created pathways that are created by the cell's behavior remodeling as shown in B. However, so the experiment that I would set up would possibly be creating a monolayer of BT549 cancer cells on collagen and then creating a oxygen gradient in which the periphery or the outer edges of the monolayer experience normoxia, so roughly around 8.5 to 21% oxygen. And the center of the monolayer experiences hypoxia, which would be roughly around 1% oxygen. And quantifying the difference between enzymes involved in remodeling, including LOX, which is involved in cross-linking, and MMP, which is involved in degradation, and understanding how these oxygen concentrations affect the expression of these various enzymes. Additionally, the experiment should be reproduced in 3D to better capture complex physiological phenomena. And finally, we would like to finish performing atomic force microscopy and eventually perform scanning electron microscopy on our collagen samples to have some hard data to back up our collagen samples and relate the data to our images up here, as most of our collagen observations are strictly observational. They do not have any numbers to back them up. However, that is being worked on, but for the future, that would be good to perform. And that was my presentation, but first I would like to thank everyone from the Pathak lab for their kindness and their constant willingness to help. I would also like to thank my mentor, Han-Shen Yu, and my mentor, Dr. Sarah Wei and Dr. Amit Pathak, for their always constant willingness to help. And finally, I would like to thank the Center of Engineering and Mechanobiology for giving me this opportunity this summer. I had a lot of fun and it was great. And thank you. Any questions?
How do you go about creating like that?
How
do you control the environment?
Oh, are you saying in our experiments or the future experiments?
Future experiments.
Oh, so my PI sent us a paper. It's very experimental, but we can kind of create kind of like spheres almost that deprive the collagen of oxygen. It's very experimental and it hasn't really been recreated. So it it definitely needs more testing before we actually perform it. But other than that, I don't really know how else we could do it because it's something very hard to do. Dr. Widder?
My question.
Oh, for 3D models, we would have to like seed a layer of collagen or coat have a layer of collagen. And then I'm not so sure if the 3D model should be single cell or if it's spheroids, but spheroid migration it's very slow and it's very finicky. And many members of the Pathak lab they could definitely back me up on this. It's not as straightforward as 2D single cell experiments. And additionally, it would be interesting to see how spheroid collagen remodeling differs between single cell collagen remodeling. But yeah, for the future, given more time, we should definitely recreate it in 3D, possibly using spheroids. But more investigation has to be given into that. Question? Yeah, this experiments like that have been performed by other members of the Pathak lab, and it is shown that cells do kind of have a cellular memory in which they remember their environment and they can transfer that to new environments and show a more aggressive phenotype that can be associated with cancer. So that is an area of active research, especially by the Pathak lab. Questions? That should be it. Thank you everyone for listening.
Thanks, Ross. Good job. Thanks, Pathak Lab, for coming. Okay, our next presenter is Yuvan Chali from Yale University. He's been working in the Marcus Foston lab this summer, and the title of his presentation is AFM-IR preprocessing using a convolutional neural network. Welcome, Yuvan.
Oh, this doesn't, okay.
Okay.
Hello, everyone. My name is Yuvan. I'm a rising sophomore at Yale. So the topic of my presentation is AFMIR preprocessing using convolutional neural network. So the first question is, well, what is AFMIR? So AFMIR is atomic force microscopy based infrared spectroscopy. So the way it works is it's similar to regular IR, except we can gain nanoscale resolution, which allows us to get a more higher resolution chemical information. So we target IR wavelengths basically at the surface of the sample. And then we can run a range of IR wave numbers to get the entire spectra. And we measure the thermal expansion at the surface to detect the absorption of that wavelength. So AFMR has a variety of applications across different areas, including polymers, life sciences, pharmaceuticals, and the main benefit is that we can get high resolution understanding of where different chemicals are at any given point in an experiment or in something we're trying to understand. And so one of the issues, or I guess just an overview, AFMAR data has a lot of noise like any other kind of data. And the goal of my project is to preprocess it. So preprocessing, essentially, we're trying to eliminate sources of noise that come from the instrumentation, the environment, etc. And so on the right, we have the general structure of AFMIR data. So it's kind of, you can think of it as like a 3D data set because we have a surface, and then we have collected absorption information at different wave numbers. So that produces the 3D nature. And I guess another thing is that if you take any slice, so on the right, we see the wave number map. So that's just at any given wave number, we can see the level of absorption across the surface. And so I guess my project was divided into two main areas. So the first thing is a data dependent model, and the second thing is a data independent model. And so in the dependent model, we're just trying to demonstrate that a convolutional neural network can be effective at preprocessing fMRI data across a variety of different data sets. And then the second model, we want to show that if we train a neural network on a variety of different data sets, then that can be applied to novel data sets across the spectra of the applications that I mentioned. And I guess a few different metrics that this figure will be useful in explaining is one of the ways that we can measure the effectiveness of preprocessing as compared to manually preprocessed data is something called peak signal to noise ratio. And we have different ways of measuring that, 1D, 2D, and 3D. And that's all just different ways of like feeding the data into the PSNR calculation. Yeah, I guess the current preprocessing workflow has a few challenges, mainly that it requires an understanding of the data itself and requires like expertise in the understanding what parameters to select, specifically in the smoothing and baseline correction and normalization steps. And so this manual parameter selection is not optimal because it requires expertise. And so I proposed this new workflow for preprocessing this data, although it has a few more steps. Most of these steps are just so that we can kind of standardize the data before we put it into the neural network. And basically, fMRI data comes in a lot of different ranges. So some data ranges could be like 0 to 8, and some could be 0 to 100, et cetera. And then there's a lot of like negative values due to random kind of environmental factors. And so we can use this to eliminate the need for manual selection of parameters. So yeah, the first set of results I have is, so this is a sample relating to the bile duct. And this is one example of the data dependent model. So we trained and tested it on a very similar data set. So like basically we had one big data set and we split it into thirds, and we trained it on 2 of those thirds, and then we tested on the other third, and we can see that. So the first is the raw data, and then the second image is manually preprocessed data, and then the third is the preprocessed using the model trained on that 2/3 as I mentioned. And visually we can see that it's relatively similar in terms of the spectra, and then we can also have these PSNR values, which is just essentially trying to tell us the fidelity of the model preprocessed data. And we can see that there's like around a 7 decibel improvement for each of the methods of PSNR that we calculate. And then also for the RMSE, we can see that there's a significant decrease. So the model preprocessed data is closely related to the manually preprocessed. And then visually, we can also see, as I mentioned earlier, we can look at the wave number maps here to consider whether the preprocessing was effective. And we can see that there's a clear similarity between the final 2 images, showing that the preprocessing by the model was effective. And also the numbers at the end kind of back that up, because we can see that there's also around a 10 decibel improvement in PSNR and RMSE. And so the next slide is another sample. So this is kind of to demonstrate that the neural network can be applied to a variety of different data sets. And so, but this is still the data dependent model. So we're still training and testing on very similar data sets. So it's not, this part is not generally applicable, but it still kind of shows how effective preprocessing using a neural network can be. And so we can see all the same kind of information We can see visually it looks pretty preprocessed, and then also numerically, and then same with the wave number maps and that information about the PSNR and RMSE of those. And so finally, we come to the data independent model. And so this is the main idea of what I'm trying to do here, because this has been trained not on anything related to this data set. So it's been trained on two completely different data sets. And we can see that we still have good visual preprocessing here compared to the middle one. Numerically, it's obviously going to be less because the data set we train on is different. But we can still see that there's around a 7 decibel improvement, specifically in the 1D and 3D PSNR, which give us, and also visually with the wave number maps, we can also see good similarity between the preprocessing via the model and also manual preprocessing. So overall, in conclusion, this data independent result can show us the power of this model trained on a variety of different data sets to preprocess outside of its range and be a vital step in AFMR preprocessing without the need for manual selection of parameters. Here are some references, and then I'd like to thank everyone at the Dr. Foston's lab and Dr. Kamalov's lab for their assistance with this research. Are there any questions?
Faster is it for you? Okay.
I mean, I think it's not just the speed. I guess, I mean, for an expert, it doesn't take that long to preprocess AFMR data, but I just think that it allows like people who don't necessarily have the expertise in AFMR data to easily preprocess. And also the current way of preprocessing requires a few different like coding languages, and it's just a bit, the packages are developed in different languages, and it's just a bit finicky and annoying. So I think that's the main, it just kind of streamlines the whole process. Yeah, so I've not tested it on data sets collected by other groups, but I believe that it should have applicability there because I trained it on a variety of different kind of scalings and different like samples. So I think that it should have, and also my normalization process has a good like capability of kind of standardizing different data sets, but I haven't tested it, so I'm not completely sure on that. Yeah. Since it's been trained specifically on these AFMR data sets, I think it's more applicable there. And I think for AFMR, there's specifically like a few different like changes that we make in the way that we preprocess the data. But I think it still could be valuable in. I think the main benefit of, so normally in IR, it's like we don't have like thousands of spectra. We just have like 1 or 2 spectra. So it's not super necessary to preprocess it. Whereas with AFMR, because we have such a large data set, that's, I guess, the reason why, because there's so much variance in the spectra itself that we kind of want to normalize and standardize it before we run any measures, run any like other analysis on it. So. Yeah, so I know that there's a collaboration between the Foston and Kamlev lab about this, and I think they're applying it to NMR data sets, to SANS data sets, so that this technique can also be used in those areas as well. That's it.
Thank you, Yuvan. Okay, our next presentation is by Elizabeth Bennett from the Georgia Institute of Technology. She's been working in the Dixit lab this summer, and her title is Integrating a Mechanosensitive Reporter and a Plant Cell-on-a-Chip System for Quantitative Mechanobiology Studies in Plants. Let's welcome Elizabeth.
Hi, I'm Elizabeth. Like Patricia said, I'm from Tech. I'm a rising fourth year chemical and biomolecular engineering major, and I've been in the Dixit lab this summer working on developing mechanobiology techniques for plants. So mechanosensation is a required function in all life in plants, just as it is in animals. Cells need to respond and sense their environment in order to survive. However, plant mechanosensing pathways are less understood than those of animals. But one known phenomenon that occurs in plants is the translocation of BZIP transcription factors from the cytoplasm to the nucleus after mechanical stimulation of the cell. And this is analogous but not related to the YAPTAZ system in animal cells, where YAPTAZ transcription factors also translocate into the nucleus after mechanical sensation. They perform different functions, but they do the same translocation. So we used fluorescently tagged BZIPs as reporter systems to measure the extent of mechanical force on a cell based on that translocation of the BZIPs into the nucleus from the cytoplasm. Cell-on-a-chip systems have been beneficial to the study of mechanobiology in plant cells, as you can mount single cells or organs onto materials, and then you can manipulate these materials to transduce force onto the bound cells. But this has previously not been possible in plants because there weren't these materials that plants can bind to and grow on until recently with the development of these PVDF, TRFE electrospun fibrous scaffolds that were made by our collaborators at Columbia. They bind to the plant cell walls, and we have seen moss and tobacco BY2 cells adhere and grow onto the material. So I used these scaffolds to transduce force onto the bound cells by manipulating the scaffolds, effectively making a plant on a cell on a chip system. So combining these two techniques, I use them in Fuscimitrium patens or spreading earth moss as my model system for these experiments, as it's easily cultivated and grows very quickly, has simple genetics, efficient homologous recombination, and it's easy to image as it grows in a single cell layer. So combining the reporter system and the scaffolds using Fuscimitrium patens, we can create a quantitative system for the study of mechanobiology in plant cells. In order to do this, I measured BZIP responsiveness to osmotic shock assays, which is an internal stimulation from the pressure changes due to osmotic gradients. So you're creating a gradient outside of the cell, causing water to flow in and out of the cell, causing pressure changes. So in order to do this, various downshock solutions were pipetted through a flow chamber that I created to shock the samples. I also measured bound moss BZIP response to physical manipulation of scaffolds, which is an external stimuli through physical manipulation rather than osmotic shock. So seated scaffolds were fixed onto 2 cover slips, and then one cover slip was lifted to bend the scaffold at a certain angle. The scaffolds were folded and then immediately laid flat and imaged for every 10 minutes. For the osmotic shock assays, it was for 2 hours, and for the scaffold folding assays, it's for 1 hour. Here's an example of BZIP translocation. So you can see the cytoplasmic fluorescent signal in the 0-minute image, so directly after the osmotic shock or the down shock solution was flown through. And then 50 minutes later, you see that signal has moved into the nuclei, and it's more concentrated and brighter, and that diffused cytoplasmic signal is no longer present. I quantified this nuclear signal in 2 ways. One is by measuring the number of nuclei with this BZIP nuclear localization, and the other is by measuring the fluorescence intensity of those nuclei. So this is the quantification of that. This is the BZIP translocation in response to osmotic shocks, with the figure A showing the nuclear intensity, signal intensity, and the image B showing the number of nuclei present. So the peak fluorescence occurs at 50 to 60 minutes post-stimulation in the highest of the 3 shocks, with the highest translocation occurring in the highest shock, which is the negative 200 millimolar down shock, which is at the top. Also, intensity is artificially high at the 50-minute time point due to just the image analysis process, but when comparing the nuclear levels with the number of nuclei, you can see that for the negative 50 millimolar down shock and then the isotonic control, there really isn't significant BZIP translocation, indicating that the negative 100 millimolar down shock is the threshold to see BZIP reporter activity. I also stimulated the cells via folding of the seated scaffolds, as seen here. So image A is a out image of like a bright field image of me folding the scaffold. So this scaffold, you can see like the moss on it. This is wax holding it down to the cover slips. Line, or the dashed line, is the axis of folding. And then in figure B, we zoom in, and this is the scale of imaging, and you can see cells and the there are nuclei exhibiting fluorescent signal because of that BZIP translocation around the axis of folding. So here we can see that BZIP translocation occurs after scaffold manipulation at the 90, 135, and 180 degree folds. Translocation occurs faster from scaffold manipulation than from osmotic shock, with peak fluorescence intensity occurring 0 to 10 minutes after folding, so directly after folding, almost immediately, compared to the 50 minutes after osmotic shock. And then the signal decreases back to normal levels after like 30 to 40 minutes, again, much faster than we see in the osmotic shock assays. And we see a higher degree of BZIP translocation after higher degrees of folding, with the 45 degree folds and then the 0 control, no folding samples not showing signal. So in conclusion, I first determined a threshold for the BZIP reporter system to allow this system to be used more effectively to determine the presence of mechanical stress on plants. And because I've quantified the system, we can now use that knowledge as a baseline to investigate how manipulating various other variables can affect that BZIP translocation, both in its intensity and in its pace, compared to what we know this baseline is. I also use these novel scaffolds to transduce mechanical force onto cells to create this method of studying mechanical perturbation in plants. So before this, being able to quantifiably externally stimulate the cells outside of osmotic shock. So that's beneficial in that. And then combining these tools with the scaffolds as a way to transduce the mechanical force and the BZIP reporter system as a way to measure that mechanical or the effects of that mechanical force, we can use these tools to open doors for further research in the understudied field of engineering mechanobiology in plant cells. So I want to thank the CMBE program for having me and also the Dixit lab for housing me. They are wonderful. And I'm very sad to leave them. Thank you to Dr. Dixit and Josh, my amazing, wonderful mentor, and also to our collaborators at Columbia for developing the scaffolds. Any questions? Yeah, so like different molarities of like, I use mannitol solutions, but like varying molarities. So all the samples start out at in the same molarity solution, then they get various down shocks. Yeah, so like, know what the pathway is. We know that these BZIPs go into the nucleus after mechanical simulation, but we're not really sure of all of the upstream steps. So it's possible. Typically, I think it's known that they use similar pathways, like similar proteins and are used, but there could be some difference. Yeah, and it's also interesting, just like the osmotic shock is kind of like we flow in the down shock solution and then they sit in the down shock solution. It's not like they're down shocked and then moved back into the normal solution, whereas like the folding, like they're manipulated and then they're put back to normal. So I think that could also affect things. Thank you.
Stevens from University of the Incarnate Word. She's been in the Marcus Faustin lab this summer. And her title is Understanding Micromechanical Property Enhancement of Pectin-Based Renewable Nanomaterials by Surface Modified Cellulose Nanocrystals. Let's welcome Alison.
Hi, like Dr. Widder said, I'm Alison, and I'm a senior at the University of Incarnate Word. And my project is Understanding Micromechanical Property Enhancement of Pectin-Based Renewable Nanomaterials by Surface Modified Cellulose Nanocrystals. Essentially, we're working in an environmentally conscious approach, and our research is focused on developing biodegradable and sustainable alternatives to synthetic plastics in the food packaging industry. We're creating pectin-based films due to pectin's high biodegradability, biocompatibility, and non-toxicity. Pectin's a natural polysaccharide found in the cell walls of fruits and vegetables and can be thought of as the glue holding plant cells together. Pectin-based films are gaining popularity because they decompose naturally, which reduces the environmental impact. However, pectin alone lacks its mechanical and thermal properties required for effective packaging. In order to combat this, we're creating something called a nanocomposite. Nanocomposites, or just composites in general, are like a mixture. So if you have one thing that has a bunch of great qualities that you really want to exhibit in your final product, but it's lacking in some areas, you're able to combat that by adding something that's really good in those qualities. And it's kind of like adding filler into a matrix. In order to enhance pectin's properties, we're incorporating cellulose nanocrystals, or CNC, which are derived from the structural components of plants. And you can think of CNCs as tiny reinforcing rods that provide strength within the matrix. These nanocrystals are exceptionally strong, and due to their high aspect ratio and crystallinity, they make an ideal filler. For comparison, CNCs offer mechanical properties similar to those of Kevlar, making them much stronger than common materials like wood or regular plastics. CNC is derived from cellulosic biomass or just plant waste materials. It's a low-cost and environmentally friendly option to act as a filler. And what we're doing a little bit differently, because CNC has already been introduced in similar polymer matrices, and we've seen positive mechanical features enhanced in these systems, is we're actually catenizing the CNC as well. So what that means is we're adding positive charge on the surface of the CNC, and we expect it to improve dispersion and improve the mechanical properties due to charge-charge interactions. We expect it to improve dispersion because we're increasing the amount of charges present, and we expect it to increase the mechanical properties as well due to the positive and negative interaction, because pectin has a lot of negative charges on the surface already, and so does CNC regularly. So the negative and the positive charges will attract and create a stronger attachment between the filler and the matrix, leading to positive enhancement in mechanical properties. In order to do so, we're catenizing the CNC with a nucleophilic addition reaction, which features an epoxide ring opening. So the OH groups on the surface of the CNC act as a nucleophile, and it attaches at this carbon, which breaks this bond, opening the epoxide ring, and then a hydrogen from the sodium hydroxide attaches to the free oxygen on that, including an additional OH bond. This reaction takes about 5 hours, and then the solid's isolated through the centrifuge and a dialysis process, and then it's freeze-dried with liquid nitrogen and then dried out in the lyophilizer. And to actually make the films, we used a solvent casting method in which we took CNC or the cationic CNC, mixed it with pectin and a plasticizer. We used polyethylene glycol 1000, and we dispersed it into our film-forming solution, which was then cast. After about 48 hours, they were fully dry. They were peeled from the Petri dishes and then conditioned in the humidity chamber for an additional 48 hours before any testing was done. For analyzing the different materials, we used a couple different techniques. The first technique we used is TEM, or transmission electron microscopy. This is a form of microscopy that looks at the nanoscopic level, and this is CNC dispersed in an aqueous suspension to get a better understanding of not only the size, but also the morphological properties of the CNC itself. Like I mentioned earlier, they are rod-shaped crystals, and we don't have the TEM image of the cationic CNC yet, but we will, and we expect it to have similar morphological properties. We expect it to keep the same rod shape, as well as the surface modification is relatively small, so we don't expect it to have a huge impact on size as well. Prior to being put in the films, we also used FTIR, or Fourier transform infrared spectroscopy, which is a spectroscopic technique that basically produces a molecular fingerprint. And we did that for the CNC and the cationic CNC before being placed in the films. And as you can see, there's not a huge difference in the two spectrums, and that's because we're adding a lot of methyl groups. And what that does is it impacts the messier fingerprint region closer to beyond the 1300 wavelength. And also, like I said, it's a very small surface addition, so we don't expect it to show up much in the molecular fingerprint. We also did FTIR for all of the films produced. As you can see, we did a pure pectin film as the control, and in all of the films with CNC or cationic C and the pectin, you're able to see the defining features of both fingerprints present in all of the films that we produced. So we were able to confirm that both CNC or cationic CNC and pectin were present in the final films. We also used DLS, or dynamic light scattering, to characterize the charge differential between the cationic and the regular CNC. And we did this by determining the zeta potential, which is essentially the voltage on the surface of the CNC or the cationic CNC, and we were able to see a positive increase with the cationic CNC. So we were able to determine that we did change the charge on the surface and we made it more positive. We also were taking a closer look at the elemental analysis of the CNC and the cationic CNC through XPS or X-ray photoelectric spectroscopy. And essentially to do this process, the surface modification can attach to the CNC or react with the CNC in 3 different ways. It can either not attach at all, be linked through weak interactions like hydrogen bonding or ionic interactions, and be covalently bound. We're looking to isolate just the covalently bound surface modifications. So in order to do that, we used a socklet extraction and we washed both the CNC and the cationic CNC with ethanol and water to isolate just the covalently bound surface modification groups. And we were able to see an increase from 0 to 0.9% of nitrogen content on the surface of the cationic CNC. And because we're adding the quaternary nitrogen, that is the element we wanted to isolate and confirm the presence of in the cationic CNC. Finally, to test the mechanical properties of our films, we were looking at ultimate strength and Young's modulus. Ultimate strength is the overall strength properties of the film itself. And as you can see at the 2.5% loading, we were able to see a statistically significant increase between the CNC and the cationic CNC at 2.5% loading. And while there was an increase at the 5% loading as well, it was not deemed statistically significant. And when looking at Young's modulus, again, you were able to see an increase that was statistically significant at the 2.5% loading. And while there was an increase again in the 5% loading, it was not deemed statistically significant. As a recap of all of our results, we were able to characterize the CNC and the cationic CNC using TM, which we were able to look at the relative size and the morphology of the CNC. We were able to confirm that there was an increase in zeta potential, which confirmed that the cationic CNC carried a more positive charge than regular CNC. We also used XPS to confirm the presence of nitrogen, which was included in the surface modification. And then after conditioning, the nanocomposites were characterized with FTIR and the mechanical testing. FTIR showed little difference between loading and presence of surface modification, which was to be expected. There was a significant difference between the CNC and the cationic CNC at the 2.5% loading. And we believe it's due to the charge-charge interactions between the cationic CNC and the pectin itself. However, there was not a significant difference between the CNC and the cationic CNC at the 5% loading, and we likely believe it's due to the aggregation of the CNC. And in order to confirm this, we will be taking TEM images of all of the nanocomposites as well. The uniform dispersion significantly boosts the mechanical strength and stiffness of the films, as demonstrated by our preliminary test, showing a notable increase in ultimate strength and Young modulus at just 2.5% cationic CNC loading compared to the 2.5% non-cationic CNC loading. However, we still need to test for permeability and water absorption to ensure these films meet all practical requirements for food packaging. This research is a step towards more sustainable, environmentally friendly packaging options. Anyone have any questions? Well, the difference with that is because these films are like very thin. They're very not like thick plastics, and they do take a long time to produce thicker plastics like that, that it wouldn't be advantageous to try and produce like the thicker, more sustainable practice. These are more like a one-time use biodegradable option that reduces plastic waste, whereas that's more of like a reusable plastic, and this wouldn't be suitable for long-term use. Yes. Yes, so CNC is biodegradable. It is derived from plant-based materials. So all of our materials are plant-based materials, and the CNC is actually isolated from paper pulp and can be isolated from plant or cellulosic biomass or bio-waste. So it makes it a cost-effective option as well as being biodegradable.
Thank you, Alison. All right, our next presenter is Stephen Wheat from Louisiana State University. He's been in the Wagon Seal Lab this summer, and the title is Piezo-1 Activation in Vascular Smooth Muscle Cells Leads to Alteration of Synthetic Gene Expression. So welcome, Stephen.
Hello. All right. Good morning, everybody. My name's Stephen. I'm one of the summer interns in the Center of Engineering Mechanobiology. Over the past couple of months, I've been working in Dr. Jessica Wagenseal's lab, studying mechanosensing in arterial disease. And today I'd like to present my poster titled Piezo-1 Activation in VSMCs Leads to an Alteration in Synthetic Gene Expression. So more on the next slide as to what that exactly means. That's really funny. Okay. All right.
Now click that.
Okay.
No.
I'll just stay
Alas.
here. We take it as it comes. All right.
Great.
So in recent years, Piezo-1 and Piezo-2, which are mechanically gated calcium channels, have been heavily investigated within the context of vascular mechanobiology. Piezo-1 specifically, which is what I'm interested in, has been heavily implicated in stenosis in aortic aneurysm. And a couple of papers that I read show that overactivation leads to vascular remodeling in abdominal aortic aneurysms. Also, overactivation leads to impaired mechanosensation. So a bit more specifically on that, Piezo-1 activation is a prerequisite for MMP3 expression. MMP3 is an upstream matrix metalloproteinase. It is a matrix remodeling marker. And so what that means is it degrades certain ECM, and it's upstream in the sense that it'll activate other MMPs, namely 1, 7, and 9, which can expand the scope of native ECM that can be degraded. Overactivation also leads to an upregulation of inflammation and senescence markers, and causes an increase in cell contractility and cell stress from high intracellular calcium levels for a prolonged period of time. So we hypothesized that Piezo-1 overactivation accelerates vascular remodeling in arterial disease by sort of transitioning vascular smooth muscle cells to a more synthetic phenotype. And sort of what I mean, back to what a synthetic phenotype means, it means that the behaviors that these smooth muscle cells are exerting they have higher rates of proliferation, and they have increased behaviors and ECM remodeling and ECM deposition. And so some of the genes that I wanted to check via qPCR are indicative of those behaviors. So CTGF is connective tissue growth factor, and so that's really involved in increasing proliferation of smooth muscle cells and increasing deposition of collagen. If you think of collagen as a structural protein that can increase the rigidity of tissue and compromise arterial function and reduce elasticity, which you need to smooth pulsatile flow. And MMP3, as I've already mentioned, and also collagen 6 alpha is one of those matrix stiffness markers because I'm interested in vascular remodeling and maladaptive vascular remodeling. So moving on to sort of what I did and why I did it. So I included this diagram that I made. It's largely based off of another figure that another paper did on osmolarity and sort of piezo activation in chondrocytes as modulated by changes in osmolarity. And sort of what I'm trying to show here is with hypoosmotic conditioning, you think the cell swells, the membrane is more taut, more piezos are exposed for activation. And you think hyperosmotic conditioning, which we did about, I believe, 500 milliosmoles for hyperosmotic conditioning, 100 milliosmoles for hypoosmotic conditioning, so about 200 off baseline, which is about 300 milliosmoles. But so in hyperosmotic conditioning, you think the cell shrinks, there's folding of that membrane, and there are less piezos exposed for activation. And since we were interested specifically in piezo 1 and no other mechanically gated calcium channels, we used a specific agonist, namely Yoda-1, to stimulate piezos. So next step, sort of to confirm that, number one, osmolarity is sort of changing the expression of piezos. Number two, that Yoda-1 is acting as an agonist. We conducted some real-time calcium imaging. So we stained MOVAs, which is a murine smooth muscle cell line, with fluorophore and fluoro-red dyes, red indicating low intracellular calcium, green indicating high intracellular calcium, and then we osmotically loaded these cells to hopefully induce expression of mechanically gated calcium channels. And then we stimulated with Yoda-1 at about, I believe, 100 micromolar and conducted real-time calcium imaging to see expression over time. So following confirmation of those pathways, we could conduct a chronic study, so see how prolonged piezo-1 activation actually affects downstream signaling in smooth muscle cell expression. So sort of to describe this, so we osmotically loaded cells for about 3 days, about 1 hour each day, at about 0.1% DMSO vehicle, which is what the Yoda-1 is dissolved in, or 1 micromolar Yoda-1. And we did IL-6 on the media before and after, IL-6 ELISA on media before and after to gauge an inflammatory response. And we extracted RNA about 24 hours after conditioning so that we could determine sort of expression of MMP3 and downstream remodeling markers. And then finally, about last 2 weeks, we started some ex vivo study on a wire myograph. And sort of what that involved was mounting a 1 millimeter aortic ring from a C57 wild type mouse, inducing a controlled stretch and stimulating with about 100 micromolar Yoda-1 or respective volume vehicle. So on to some of the fun stuff. Here's some pretty pictures. So on the right, you can see some images of our cells following conditioning. So we took an ISO baseline in about 20 frames, and then we osmotically loaded these cells at about hypo or ISO for about 10 minutes at about 0.33 frames per second. And each of those pictures represents sort of the end of that conditioning stage. And then following osmotic loading, we stimulated again at about 100 micromolar Yoda-1 for 10 minutes, 0.33 frames per second. And it's a bit hard to see. I mean, the point of real-time calcium imaging is just you can see change over time. So static images don't really do the job. So we have this quantified in MATLAB on the left in RFU. So that's change in green to red fluorescence. So we can see the increase represents an increase in calcium flux. And you can see in our hypoosmotic conditioned cells, there is more of an increase than in our ISO. But then after we add Yoda-1, you can see that little divot once we add, and we had to readjust and refocus the confocal microscope on the cells. You see a sharp increase to this maximum RFU value. For the hypoosmotic cells, an ISO is a bit slower increase in this RFU. It looks a bit more logarithmic. And so this means sort of a number of things. So the maximum, I think, might have to do with I mean, the piezos are saturated with Yoda-1 since we are adding such a high concentration. And sort of the very drastic increase in hypoosmotic cells could mean that there are more piezos available because the membrane is taut. Those piezos are more exposed. So they are more ready for Yoda-1 stimulation. So following this confirmation, we ran, again, our chronic studies. So here's some of the qPCR data. On the top left, these cells were only osmotically loaded. We didn't add any Yoda-1. We didn't add any vehicle. But B through D, we do either add Yoda-1 at about 1 micromolar or DMSO vehicle at about 0.1%. And you can look at some general trends here. We do need a larger sample size to run a more robust set of data analyses. But you can see some general trends. So like for CTGF expression, as we decrease osmolarity, we do see a trend towards increasing. We can't quite call that significant with this sample size, but you can see a general trend towards increasing. And again, whenever we osmotically load the cells, we're not really targeting piezo 1. We are, but we're also, it's not very specific. We're targeting all mechanically gated calcium channels. And then you can see for CTGF expression, this is really pretty interesting. Whenever we get to CTGF expression and MMP expression with Yoda-1 or with vehicle, you can see like whenever it's not at baseline, whenever it's hypoosmotic or hyperosmotic, you can see a more discernible change or increase in expression with Yoda-1 stimulation, though at baseline, at ISO, there is really no discernible change. So it's hard to say whether or not this is, these expressions are affected by piezo expression. It could possibly be an increase in cell stress, though further testing needs to be done in order to sort of name these significant and further investigate these claims. And here's just some of the IL-6 quantity data. So I normalized this to a IL-6 quantity before conditioning. So I took 500 microliters from my cells before I started osmotically loading and stimulating with my agonist. And then 24 hours after, I took media again, just for all downstream signaling to occur. And I quantified that using the standard in picograms per mil. And here, this is quantified in R as amount after over amount before. And I ran a two-way ANOVA, and I found that there was a significant difference between, bless you, there was a significant difference between ISO and hypoosmotic groups. There was not a significant difference between drug trials, though. So that could possibly implicate that the exposure of piezos does have sort of an effect on inflammatory response, though, again, I'd like to do more testing, but unfortunately, this REU ended. And so finally, here's some pretty graphs demonstrating some of that myograph study. So what we did is we took 1 millimeter aortic rings. We mounted those to a myograph. We induced a controlled stretch, and we added either Yoda-1 at about 100 micromolar or DMSO at about 10%. So this is really pretty interesting because you can see I've got a loud voice, I can step away from the microphone. You can see with our vehicle, there is this relaxation of vascular smooth muscle, though it's not. So that is very interesting. You can see Yoda-1, since again, that is dissolved in DMSO. There is a decrease in baseline. However, that decrease in baseline contraction is significantly less, as you can see in the, I believe that's graph B. That's the difference between baseline 1 and baseline 2 for those two Yoda-1 trial and the vehicle trial. And it's a bit hard to see because the scaling is so large, but there is a peak in contraction, and that is quantified in graph C. So you can see that Yoda-1 trial following first round of stimulation. And then when we move on to the second round of stimulation, you can see a lot of these responses in the Yoda-1 trial are blunted, and that there is a very insignificant, pretty much almost no change in contraction. And if we're talking about measuring that peak, there's really no peak in that second round of stimulation. Maybe, I apologize, the numbers are a little bit small, but the scale on graph D is very small. So that could just be pipetting. The force transducer picked up some of that because the force transducer for the wire myograph, since we're measuring contraction of such small tissue segments, it is very sensitive. So this would be interesting to run further. This is with a sample size of 2 for control and for Yoda-1 stimulation. So furthermore, here are some discussions in future works. This really recapitulates a lot of what I've already said and a lot of my slides. So the Salvatore ETL, that was sort of the paper I discussed earlier on osmotic loading in chondrocytes and how that causes variation in piezo exposure. And a lot of our imaging results confirmed that, in that we did see a much faster increase in intracellular calcium with real-time calcium imaging for hypoosmotic conditioned MOVAs, which indicates that they were more available for YODA1 activation. And RT-qPCR results suggest that changes in synthetic gene expression might be dependent upon the availability of piezo1. However, further work is needed to support this claim / investigate this claim. And again, I already discussed on the IL-6 that there was a significant difference between the iso and hypotonic conditioned cells. So testing of synthetic phenotype markers as well as inflammatory markers in response to piezo1 warrants further investigation. And a lot of that blunted response that y'all saw on the slide beforehand could indicate that there is sort of impaired mechanosensitivity following that first round of piezo activation. So I'll sort of complete this with a nice little statement. So yes, the study of piezo mechanosensation presents a novel perspective for investigating matrix remodeling and changes in cell behavior in arterial disease. And I'd like to give some thanks to my lab, also some of the papers I read. I'd like to thank the NSF, I have to. I'd like to thank the NIH. And I'd like to thank a lot of members of my lab. I'd like to thank my bench mentor for being so wonderful, Dr. Garrett Eason. I'd like to thank my lab PI, Dr. Jessica Wagenseal, for meeting with me every week. She was always very available, and I really greatly appreciate that. I'd like to thank all the members of my lab. I'd like to thank Viola for taking a picture of me during my presentation. I'd like to thank all the undergrads for providing great company. Okay. Here's my poster. I'm finished. Questions? I'm sorry. Right now. A lot of the focus on the RO1 is on piezo2, especially because they did run some single cell RNA-seq and they did find peaks in human thoracic aortas in piezo2. So, but yeah, so piezo2 is the main focus in the lab, but I got piezo1, so that's what, that's what I've been largely focused on. That's what I've been reading on, and I've enjoyed it. Any other questions? No one? No. Okay.
Okay, thank you, Stephen. It's on, yeah. Oh, you can carry that. Is it just the poster?
Yeah, it's just.
That's fine.
Excited.
That is okay.
It's kind of ugly, but it's okay. It's kind of ugly. It's gonna go be beautiful one day.
Oh, you're doing just fine. Okay. Our next presenter is Huamin Gao from the University of Illinois at Urbana-Champaign. She's been in the Fuzhong lab this summer, and her title is Heterologous Expression of Engineered Titan Immunoglobulin Domain Mutants for Sequence Property Relationship Analysis. Let's welcome Huamin. Sit pointer in here, sure.
The microphone? Oh God, that's terrifying. I don't like listening to my own voice. Okay. I'm gonna hope that doesn't fall. Or I must put it on the ground. All right. Hello everyone. My name is Famin, and does the microphone hear me? Okay, that's true. Okay, hello everyone. My name is Famin, and I am a rising senior at the University of Illinois, Urbana-Champaign. I am, as Dr. Widder mentioned, in the Fuzhong Zhang lab this summer, working on a project for the heterologous expression of engineered Titan immunoglobulin domain mutants for sequence property analysis. So for some background, what is Titan and why is it important is the big question that I'll be answering. So Titan is one of the three muscle fibers found in your skeletal muscle. It is also the largest one out of the three. And Titan has a lot of properties. It contributes to your muscle's property of elasticity and as well as like structural integrity and a lot of that stuff. So for this reason, because of these properties, it's been seen as a promising material for a lot of biomedical and like industrial applications. So for example, it could be used as like scaffolding for muscular like regeneration or something like that, as well as like drug delivery and a lot of other applications. So because of this, there has been previous work done on it. So Titan gets its properties from things called immunoglobulin domains, I always get that wrong, which are basically like monomer units of a larger polymer. In Titan, I think there's over 200 different immunoglobulin domains, and the structure of immunoglobulin domain, I don't have an image on it, I kind of wish I put one in, but it's basically a beta sheet sandwich that is comprised of like two beta sheets squished really close together. And each of these beta sheets consists of around 3 or 4 beta strands. So that's the general structure of the immunoglobulin domains. The previous work that has been done on Titan focused on 4 specific immunoglobulin domains that were randomly chosen. We call these IG domains 67 to 70. And previous work has gone through the process of first expressing the protein and then large scale production, getting the protein powder and then spinning a fiber out of it. And this fiber was used for mechanical testing, and it was found that the mechanical properties were actually quite good. So more work is being done on Titan now. Currently, the project is trying to figure out the sequence property relationship between Titan's sequence and its mechanical properties. So the first step in actually trying to figure out what this relationship is to actually express the protein in the E. coli bacteria that we use in our lab. So we're trying to produce it through a synthetic biology pathway, and the first step to doing that is expressing it. So before all that, the method that was used to actually get these mutants was a computational model was used in order to predict what kinds of mutations would result in what, what results. And so the computational model would take one of the native Titan IG domain sequences, and it would introduce mutations to it, and it would say whether or not it results in a better mechanical properties. So after we obtain these sequences, we would clone them into a plasmid backbone, which consists of an antibiotic resistance. And after we would do that, we would transform them into the E. coli strand NED10 beta. And after transforming, so we would plate it onto an agar plate and then let the colonies grow. As you can see from this thing right here, this is just a schematic of the basic workflow of what I did this summer. So after we transformed it, there are, I think, 3 steps that we would take to actually get the protein powder. The first one is to check that the proteins actually express. And we do that using an SDS page gel. So the process goes more or less like this, where you first seed one of the colonies into 50 milliliters of Luria broth media, LB media. I'm sure you heard Abria talk about it earlier. And after we let it grow overnight at 37 degrees Celsius, this 1 milliliter of that sample was then put into terrific broth TB media to grow for around 10 hours or until the optical density or OD reaches a value of 3. So after the optical density reaches a value of 3, the sample was collected, an uninduced sample was collected, and the sample was induced. So what induction basically does, it just encourages the production of the protein. So theoretically, if the expression goes well, you should see like a brighter band around where our protein would be located. And I'll talk about that later in our results section. So after we confirm the expression of our protein, we would then go on and talk about the large scale expression or like the large scale production of our protein, because in the end, remember, we want to obtain a protein product so we can actually spin it into a fiber and get the mechanical properties. So how we got the protein powder is we used large scale. So it's very similar. The start of it is at least very similar to the small scale expression, where we first start off, instead with 50 milliliters LB, we start off with 150 milliliters LB, because we end up wanting to seed it into like around 10 liters to produce like all of our protein, because we need a lot. So after the 150 milliliter LB culture is finished growing, we leave it grow overnight. Again, we put it into TV media. This time we put it into 1 liter TV media and a 2 liter flask. And we basically let grow again over 10 hours or until the OD reaches 3. And then afterwards, we induce it, collect the sample, stuff like that. And that process is basically similar, but instead of getting rid of the cultures afterwards, after we collect the samples, this time we do collect the cultures, and we centrifuge it and collect the cell pellets, because that's where all your proteins will be, because your cells are not lysed yet. So the media doesn't have anything, but the cell pellet does. So after we collect the cell pellet and get rid of the media, we then go ahead and lyse the cells using guanidine HCl buffer, which basically just breaks the cell apart. So then your protein will be in the lysate instead of like the cells. So then afterwards, we centrifuge that again, get rid of the cell pellet this time because your proteins are not there. And then we run that through my favorite part of the entire summer, purification columns. So purification columns, we use a nickel ion column, and we use 300 millimole imidazole to elute it. So how the nickel ion column works is our proteins will have a his tail or like something at the end, which makes it really, really sticky to the nickel ions. And the other proteins have shorter tails or whatever, so they'll be less sticky. So basically, we first wash the protein with imidazole dissolved in a urea buffer. We first do 20 millimoles and then 50 millimoles. And then after the column is washed, we then go ahead and elute it using 300 millimole imidazole. So then after this elution, the proteins are then dialyzed in first 5% acetic acid and then 1% acetic acid. They are freeze-dried and then weighed to see how much protein powder we actually got. And then they're stored for later mechanical testing at -80. So that process is basically numerically described by this diagram. And then for results, we were able to get, at least for cloning the sequence into the plasmid, we were able to get quite a few that we all verified through sequencing. But among those, we got 4 type mutants to successfully express in E. coli. Sorry, I'm like not speaking to the mic. So as you can see from this diagram right here, this is one of the type mutants that we ran on an SDS page gel. Here's the ladder. Our protein is found between 37 and 50 because it is around 44 kDa. So this well represents the uninduced sample, and this one represents the induced sample. So you can see at the uninduced sample at 44 kDa, there's not really anything, but in the induced sample, you can see that there is a much brighter and more intense band, which means that our protein was expressed and it expressed a lot. So this is just checking expression of the protein. So after we confirmed the expression like this, we ended up going through with large scale expression, and of the 4 type mutants, we had an average titer of around 50 milligrams per liter. And these are the results of like the purification of that protein. So you can see that as opposed to like this, like the uninduced and induced samples over here that aren't purified, you can see that this is a lot of protein, and it's also pretty pure because there's not really any other bands on the same gel. So this is the protein that we obtained from, I guess, the purification process with the nickel ion column. So what does this project mean, and what is the significance? So the significance is that we ended up being able to express Titan mutants So that means that the, I guess, protein powder that we obtain can then be used for further mechanical testing, because again, the aim of this project is to figure out a relationship between the sequence of your Titan immunoglobulin domains and their properties. So the protein powders that I made over the summer will then be used for like mechanical testing on the tensometer, and hopefully a sequence property relationship can be established. So expression also allows for, I guess, a more sustainable production through a biological pathway. So because we can, like, I guess, figure out the, hopefully figure out the sequence property relationship, and once like we understand that process and like what the relationship is, we can then go ahead and tune Titan's sequence to like the mechanical properties, and hopefully it can be tuned in a way to be actually useful for a lot of different applications. But yeah, that's my poster so far. That's my poster. I'd like to thank everyone in the Zhang lab for being very supportive and great. And I'd also like to thank CEMB and SMARC for funding and supporting my research. And I'd also like to give a thank to my mentor, who's sitting back there, for teaching me a lot of things. But yeah, any questions? Oh. Yes.
So this one.
So the 67 to 70 was done in a previous like paper done by the lab. 67 and 70 are like different immunoglobulin domains, but I think for the purposes of the project, we use, I guess, like one of the native ones that were studied by the other paper.
The reason I'm asking.
Mm-hmm. I think what it is I guess if you think about like a polymer chain, so it's like one of the IG domains is like one of the units of the monomers, so that it gets connected into a large chain, and that's what is like the fiber, I think. Yeah. Something like that, I think.
Our next presenter is Eliza Mujahid from the New Jersey Institute of Technology. She's been in the Guygenin lab, and her title is Understanding Fibroblast Activation and Cardiac Fibrosis, a Cluster Model Approach. Let me get rid of this. Thank you.
I'm just going to check how this thing works. Okay. Well, hello, everyone. My name is Eliza Mujahid. I'm a rising senior in New Jersey Institute of Technology. My major is biomedical engineering. So this summer, I've been working in Dr. Guygenin's lab. So over this summer, my research was focused on understanding the fibroblast activation in cardiac fibrosis. And for that, we basically had a cluster model approach. As a background to why understanding the fibroblast activation is important, it's because every year there are 800,000 plus deaths that's caused by either cardiac fibrosis or lungs fibrosis. So what is basically fibrosis? Fibrosis is the activation of fibroblast cells. Those are certain type of cells that are present in our heart, and they are transformed into myofibroblast cells, and they are sticky in nature, which causes a mechanical and biochemical change in the cells. And that leads towards the heart function to alter and be abnormal because it starts to stiffen that heart cells. And the driving factor that we believe is because of cell clustering. Now, what is cell clustering? It's basically when the cell starts to get together and stick in a specific region of the heart. And that leads towards the cell and the heart itself to stiffen. In order to have the study, we had a theoretical approach and we had an experimental approach. where we investigated how cell density, like how varying the number of cells, as well as how pharmacological would interact with the cells to alter the cell clustering. As you can see, we have our theoretical model over here. So N0 is basically normal number of like fibroblast cells and myofibroblast cells present in heart. So as the nucleation rate increases, the number that N1T shows that the myofibroblast starts to cluster together in heart. And what basically happens is that once there's like a myofibroblast in heart, the surrounding fibroblast cells start to cluster together. They come around, which is basically the factor present over here, which further increase the clustering. So as it gets bigger and bigger, further cells around it also start to get attached to the myofibroblast, and the clustering happens, and the ECM, which is the extracellular matrix, it starts to stiff more and more, and the heart function alters with it. So with this predicted data, then we did the experimental data. So we basically tested out two things in our experimental procedure, which was one, as I mentioned earlier, we had two different cell densities. One is 0.3 million cells per ml, which is over here. And the second was 0.5 million per ml of cells in there. The second thing that we tested was we wanted to check that how does microtubule polymerization would affect the cell clustering. So this images that you see is what we did for. So how I started up the methodology behind doing all of this procedure was first I cultured the cell in a collagen matrix. Then I used 0.3 millions per ml cells in it. And with these, I can see the two experimental data, which I will tell you guys later. And for the second part, I used in order to test how microtubule polymerization was affected, I used noctazole drug, which is basically a drug that's used to inhibit the microtubule polymerization. So I had three steps. One is pre-treatment, control, and post. Pre-treatment is when I added a drug in the start. In control, I didn't add the drug at all. And in the post-treatment, I added drug 18 hours later, and then I let it stay for 6 hours so that the whole 24-hour duration was completed. And after the whole 24-hour duration, I had all of these three different sort of cell cultures to be tested in the confocal microscopy. So these images that you see are basically confocal microscopy. So now moving forward towards the results, the green spot that you see is basically the collagen formation. So we depicted that as the number of cells increase, the collagen clustering also increased. So you see that there's more collagen clustering happening in the confocal microscopy, and it aligned with our experimental data. Like over here, we predicted that there seemed to be two clusters formed, and it was what we did the experiment. It was like the same way that we predicted. And when we increased the cell density to 0.5, we predicted that there was going to be like 3 collagen clusters, and that's the same thing that happened with the experimental data as well. Going forward towards the nidazole drug that we used, which was basically what we hoped that would inhibit the microtubule polymerization that is causing the cells to cluster. When we compared the post-treatment with the control treatment, we saw that there was a higher collagen clustering. And when we compared the control with the pre-treatment, we saw that the pre-treatment had a lower collagen clustering. And it was also according to our data. As a result, as a conclusion, we came to know that our study with collagen clustering and with the microtubules, that they did affected the collagen to have more clustering, which leads towards heart to become more stiff. The reason why it is an important study was now that we can use the same methodology if we inhibit these microtubules to form further, that would basically lead towards fibrosis. We can use this to have a therapeutic use and use drugs related to it, and probably leads towards cure of fibrosis. Thank you. That's my presentation. That's my poster. In the end, I would like to thank Dr. Guy Janine, my mentor Kenny. Both of them are in China, none of them are here. And my fellow undergraduate students, like I was glad meeting you all. It was a really fun summer. And thank you so much for the CEMB faculty, for NSF funding and everything that you guys have done so far. I have never done this sort of but lab stuff, so it was my first time, and this experience is truly valuable to me. So thank you so much. Any questions? Yes.
Are you using Paxil?
So we basically we have done more further thing, but we didn't use that specific drug. We have used Cal-A, we have used thrombin. Cal-A didn't give us the right thing that we desired. Thrombin did. So we are still testing more drugs on it to see how microtubules are acting with the drug itself. Any more questions?
That concludes the presentation portion of our morning. We want to close today with some final remarks from Dr. Dixit and some thank yous for the bench mentors that made this so meaningful, and so for their support of our students. I don't know what to do about that.
I think you can.
Okay.
Yeah. Okay, well, thank you to the students for wonderful presentations. It was really nice to hear what each of you did, and it was amazing to see the breadth of research projects that you guys engaged in as a group this summer. So this year, for the first time, we are doing a Best Bench Mentor Award, which I think is a wonderful idea. So a really nice way to recognize and reward bench mentors who had a big impact. So this year's inaugural winner is Josh Kumi. Yeah. So. And I'm not going to read, the nomination letter was extensive and had lots, that's a good thing, and had lots of things to say. So I'm going to just read one little snippet. That said, your mentee, Elizabeth, said that, from facilitating her growth in the lab to asking about time in the program to giving great recommendations for weekend activities, which I gathered numerous of your suggestions were actually acted upon and led to a fun time, that you did an outstanding job helping her grow professionally and personally and making her time enjoyable in the lab and in St. Louis. So congratulations to Josh. If you could step up to the podium here. I have a certificate for you here, and then your award also comes with $100 payment that you'll get as part of your paycheck next time. Congratulations. Yeah, good job. Yeah. Do you want a photo? Maybe you should get